Burst off the world, they call through the rivers that rise and fall, echoing soft through canyon walls, whispers of the wild. Raindrops play a soft marimba, dancing leaves like a green samba. Moss carpets lay a velvet trail, guiding feet through the wooded vale. Whispers of the wild, they call through the rivers that rise and fall, echoing soft through canyon walls. Whispers of the wild. Fox's shadow in the light, an owl's cry breaking the night. The earth breathes deep, a steady song, and carries us all along. Whispers of the wild, they call through the rivers that rise and fall, echoing soft through canyon walls. Whispers of the wild.
The trees hum low, a secret tune, beneath the light of a silver moon. Branches sway in time with the breeze, a quiet.
Good morning, ladies and gentlemen. You are welcome back to our online lecture that has to do with microbial evolution and ecology. This course actually started some months ago, and we were able to look at one or two things about microbial evolution. And today again, we want to actually start from here, commence by talking about our focus on microbial classification, identification, characterization, and today our topic is going to be identification, characterization, and classification of microorganism. So where we are expected to explore our understanding, our knowledge on microbial evolution, ecology, and use it as a kind of ethical implication for medical diagnosis and treatment. I want to say this, that as a medical or paramedical personnel, as a microbiologist, um, you will be dealing, there will be some points in time, especially some of you that will go into medical microbiology, there will be some time that you need to collect human sample, deal with human sample, So that has to do with collection, that is starting from the field down to the laboratory. Then for some of us that we also want to talk about dry lab, that is in silico, that is we will be able to do what is bioinformatics, you'll be able to make use of it. So today, just without wasting our time, in the next few minutes, we'll be looking at, you'll be coming across these words, these terms, you'll be coming across them one after uh, so often in this course. And the first we have to look at is horizontal gene transfer, what is called the HGT. Horizontal gene transfer refer to the transfer. Now, don't forget that I told you that one of the survivability um, strategy of any microorganism is ability to adapt into a new environment, and that has to do with changing the molecular pattern, changing the molecular mechanism in which this microorganism will have to exist in its environment. That means under normal circumstance, and don't forget that I told you that a microorganism, they do not live in isolation, they live in community. The same way you also live in your own community, that you can move from one community to another, and in a community you have different diverse you have diversity. That is, the bacteria will have to coexist with fungi, we have to coexist with other viruses and some other. Apart from that, there should be a complete interaction between the microorganism and its environment, and that has to do with a lot of mechanism are involved in this. So that is to tell you that one of the way that they do interact and communicate with each other. Some of us will not know that the same way we also interact with each other in our community. Microorganism, all cells, let me tell you that all cells, they crosstalk, they have means of communication, they have medium of communication, they have their own language. For bacteria or for any cell to undergo cell division, something must have communicated with it in order to be able to know when to get into cell division. So that means genes must continually, hereditary materials, genetic material must completely have to be transferred between one microorganism to another. So the way that is carried out, number one is through horizontal gene transfer. So horizontal gene transfer refer to the genetic material between organisms in a manner other than the traditional way. So what we are saying in essence is that at this point you need to know that the normal thing we know before of a way of transferring movement of genetic material from one organism to another can either be through transformation, can be through transduction, or it can be as a result of conjugation. But nowadays, when there is now a transfer of genetic material between microorganisms of the same species, horizontal, of different species, for example, when you have a gene transfer between E. coli and Pseudomonas, that is called horizontal gene transfer. But vertical has to do with when there is transmission of genetic material from the parents to the offspring. That is what? It is called what? It is called vertical. Another way you can also understand the difference between vertical and horizontal is that horizontal, you can also get there is some material, genetic material can also be transmitted from one organism to another as a result of environmental changes, including antibiotic resistance. So that does not in any way similar to what is obtainable in vertical gene transfer. So vertical is from parent to offspring, horizontal is from what? The environment, as a result of the environmental factors, which has to do with sharing, exchange of genetic material between organisms of either the same or different species of microorganisms. So, and the third one has to do with transformation. Transformation is when you have um, genetic exchange, when a bacteria is acquiring a foreign DNA from its environment. And transformation has to do with, okay, for example, when let's say where you have a laboratory discharge, let's say after culturing microorganism, after this, so the way you now discard the microorganism already is dead, you now discard it into the environment. What is going to happen is that the microorganism is dead, but the genetic material, the DNA, is going to be in form of a naked. It's going to be naked. And the same way that there is going to be etching. So that means the living one can also do that. Let me see, let me try and generate structure, a diagram for horizontal, for transformation. Okay, generate generate um an image to to explain transformation, um to explain transformation, transduction, horizontal gene transfer, um vertical gene transfer, as well as what, okay, in bacteria. Let us be specific. So let's ask ChatGPT to do that for us. So it's going to, so while it's doing that for us, we'll continue explaining. Okay. So the first thing is that, just like I said, okay, about identification, characterization, and classification of microorganisms. So what we are saying in essence is, in an hypothetical environment, in a hypothetical situation, whereby you don't forget that microbiology is the study of microorganisms, which include bacteria, viruses, and algae, and all this one. And I said that there's a way they normally coexist. They must exist in the community. They exist in the community. That means if you want to, as a scientist, if you want to identify them, if you want to deal with them, if you want to study them, you need first of all to isolate. You isolate. Isolation means that you now pick them from the environment. And in an attempt for you to pick them, that means you are trying to study them through a specific sample. So it's just like in any community of microorganisms, whereby you have viruses, you have bacteria, you have fungi, you have protozoa, you have algae. Before you'll be able to identify that this one is a bacteria, this one is a fungi, you want to isolate them. You want to, it's just like somebody who is looking for favor, favor martins, in HND2. The first thing is that in among the, I mean, in the population of people, I mean, students in the Okeogun Polytechnic, in that population, you have to single them out. That is, you have to go to, that's where you now ask for the matric number, you ask for the level, the course. So until when you now still get to HND1 microbiology. So if you now want to know who favor is, that means you have to isolate it. You have to isolate it. And apart from isolation, the next thing is how can you characterize them? Characterization before you be able to do. So what I'm trying to say is just that you should know that in biology, so let's see this I want to pick anyone. I've asked ChatGPT to generate this for us so that I will be able to make use of it in order to explain. So let me speak. Which one should I pick? Okay, this one is a free DNA. This one is transduction. This is vertical. This is okay. Let me pick this one so that I'll be able to make use of that one to explain the what is transformation, transduction, and horizontal gene transfer as well as a vertical gene transfer. Now I want you to look at this bacteria. Look at this bacteria. When you look at this bacteria, you will see that this is the original bacteria and this is the DNA inside the bacteria. This is the free DNA. Now, transformation now is that I told you that if this donor bacteria dies as a result of reaching the death phase or as a result of anything, it could be environmental stress, it could be maybe no more nutrient, whatever. So this DNA will now become free. You can see this DNA will now become free. This is a new bacteria now. This is a new bacteria with its own DNA already there. So if you now look at if this free DNA that is now free in the environment, this bacteria can take it up. So the exchange of genetic material between um, dead or free DNA and a recipient, a free DNA which was originally from a donor bacterium to the recipient. So if this one, let's say this one is A, bacteria A, this one is bacteria B. So if this DNA becomes so available and this recipient DNA which is B now exchange, now collect this type of DNA, it is known as a what? Horizontal, it is known as what? Transformation. That is transformation. Free or dead from dead bacteria. This one has already died. So it's like inheriting. It can be, you can explain it in when you inherit, when bacteria, a recipient bacterium, inherits a free DNA, that is known as transformation. Transform, from the word transform, that is something that is already dead. So it is now going to become active or become alive on when it gets to this recipient bacterium. So that is what? That is transformation. Now let's look at transduction. Anytime you come across the word transduction, that means a virus is involved. A virus is involved. There are some special bacteria. They are special because that is the food eaten by virus. So, and any virus that feed, whose food is a bacteria, they are called bacteriophage, or you can just call it a phage. Now, don't forget what is a virus, what a virus is. A virus is this you can see this is the head, this is the head, and these are the tails. So inside the head is where you have the DNA. So under normal circumstance, how does virus infect a bacterium? What happens is that the phage or the virus is just going to inject its own DNA. The whole virus does not take part in the replication or in the infectivity. So it is only going to release its own DNA into the system of that bacteria. And once this one has released its own DNA into this, so the phage DNA, the virus DNA will now become integrated, will now join, will now become incorporated into the main DNA of the donor. So this one is the donor bacterium, you can see the DNA, and this one is the phage. So when this phage has infected the donor bacterium, when it has infected the donor bacterium, so you can see that the DNA of the virus will now become integrated along with the DNA of the bacteria. So the exchange of genetic material between that virus and the bacteria is called transduction. It's called what? Transduction. Okay. Another one, another means by which bacteria exchange their DNA or genetic material is through the process called horizontal gene transfer. Horizontal, you can see that they are at the same level. Both the donor and the recipient, they are at the same what? The same level. It is called horizontal. So, and it's always between the parents and the offspring. It's like the um it's like hereditary. It's like the traits. It's like the traits you inherit from your parents. So that is horizontal gene transfer. And I want you to, apart from transformation, transduction, and horizontal gene transfer and vertical gene transfer, the other way by which bacteria or microorganisms also exchange their genetic material is also as a result of conjugation. What do we mean by conjugation? Conjugation is the sexual reproduction that we see in microorganisms. Conjugation, like conjugal bliss, it has to do with marriage. It has to do with sexual reproduction. So although that one is not illustrated in this diagram, but in trying to explain the horizontal gene transfer, you will see already that it is inheritance from the parent to the offspring. And I want you to also know something, that scientists later discovered that bacteria, there are certain bacteria that when you study them very well, when you closer look at them, you will see them, you will see them that they have what is called a pilus, F plus and FR. So that is, there are some bacteria that they can also be regarded as male, and there are some bacteria that can also be regarded as a female. So those ones that receive recipients are always the female, and those ones that donate are always what? Male. Okay. So horizontal gene transfer, conjugation, transformation, and transduction. I'm very sure that that one is clear to us now. So the next one is vertical gene transfer. Sorry, vertical gene transfer. Vertical is from parent to offspring. No, sorry. Vertical is from parent to the offspring. So you can see that this is the parent bacterium. This is the parent bacterium, and these are the daughter cells. The daughter cell. When you look at this DNA now, look at this DNA from the parent, you can see that some part of it are what? Are like this one is green, and this one is another color entirely. And by the time this one divide, so this one will just be divided into two, like that. So this one will now become divided, and part of the daughter cell is going to share from the parents to the offspring. So when we talk about horizontal gene transfer, that is always between um, exchange of genetic material at the same level. It could be as a result of environmental changes. It could be, for example, when let's say you introduce antibiotics into the population of bacteria, what is going to happen is that some of them will die, some will die off, but some of them will become so resistant. So the why would they become so resistant, and what are the those factors that have actually taken place for them to become so resistant? They will just go and get and acquire or change their genetic makeup. Their DNA will become, so the presence of plasmid, what we call plasmid are extra chromosomal material that you found in the bacteria. So when there is exchange of genetic material between microorganism of the same species or of different species, that is called horizontal gene transfer at the same level. At the same level. Is not vertical. Vertical, if you want to draw a vertical line, it's going to be from up to down. So when you remember vertical, up to down. And when you remember horizontal, that is left to right or right to left. It could be either. So that is horizontal gene transfer and vertical gene transfer. So another thing that I want you to know, and we are going to also take cognizance of in this course, is mutation. You come across mutation. Mutation means sudden change or sudden changes in the genetic makeup. How do they survive? When you introduce microorganism microorganism from its natural environment and you want to isolate it, you are taking it or bringing it into the laboratory, there are some genetic changes that is going to occur. And when there is a genetic changes, sudden change in genetic makeup, it is known as a mutation. Don't forget that at the end of this lecture, later on, before we have the second lecture, which is going to be anytime tomorrow or thereafter, we are going to write a quiz. There is going to be some set of questions as you normally do here. Okay, so that is a mutation. That is mutation. We have explained what is horizontal gene transfer, vertical gene transfer, transformation, transduction, conjugation, and mutation as well. Mutation. Okay. Now, having known that, the next one that we want to also explain is what is called genetic recombination. Genetic recombination. From the word recombination, recombination means that you will cut. It's like when you some pieces, okay, pieces of DNA, when you now start aligning them together, joining them together in from pieces, then to the whole DNA in no fashion. It doesn't have any order. So that's why they said genetic recombination is the process by which genetic material is shuffled. Underline that word, shuffled. Is by recombination, is by randomness. It doesn't follow any pattern. So it's shuffled between different organisms, which often increase in genetic diversity. I want you to, one of the take home I want you to know is that when we talk of evolution, there can never be anything evolution if there is no genetic recombination. There can never be evolution if there is no mutation. So mutation, genetic recombination is bringing about genetic diversity. And what do we mean by diversity? Genetic diversity, difference, different types, recombination. So don't forget that if I am talking, I'm mentioning like DNA and all this one. Whatever I say about microorganism, I want you to imagine it in their molecular senses, molecular existence. So whatever happens is that when you bring a microorganism and you culture a microorganism and the microorganism now grow, there must have been genetic changes. There must have been genetic modification. So, and that is why I want you to know that another means of exchange of genetic material is by genetic recombination. Is by genetic recombination. Another word you can also use for genetic recombination is reassortment. Sorting, random sorting of genetic information. That is that. Now, how useful how useful is your collection of samples? How useful is that to correct diagnosis and correct treatment? So don't forget that the primary goal of diagnosis is what? So that you'll be able to have a better control or a better solution so as to prevent the microorganism from that environment. The primary goal at this stage is to collect environmental or clinical sample. I told you that microorganism exists in community. They coexist with viruses, they coexist with bacteria, they coexist with fungi, protozoa, algae, and so on and so forth. So if they exist in that community, so that means if you want to isolate them from that community, the first thing is to get to collect the sample. So the sample can be environmental sample. So, for example, if you want to study bacteria or you want to study microorganism from the soil, so the soil is the environment. If you want to study that from water, the water is the environment. For example, let's say you want to look at the microbial load. You want to know the type of microorganism that you can see from food that are being sold or fended to you. You can just say, okay, oh, this fruit I am buying, I want to know the type of microorganism that are on that fruit. So the next thing is, are they fit? Those fruits, are they fit to be consumed without any problem? So, and that's why you have to, the first thing is to collect the sample. um Collect the sample. The types of sample collected will depend on the specific research. If you are going for infectious disease detection, environmental microbiology studies, for example, you may just want to know the type of microorganism from the soil, from water, from air, on the table, on your table surface, on the door, wherever that is dealing with environment. But those also don't forget that there are microorganisms that their major environment is inside human. Normally, we have some bacteria that they normally live inside in our intestine. So the types of sample you will now collect will now depend on the organism you are interested in or what you are actually interested in. Are you trying to diagnose an infection? Do you want to know the infection? So the first one is the types of sample can be clinical samples like blood, urine. If somebody is sick now and the person is complaining about headache, severe headache and all the rest. So you need to it's not that you have to tell them that, oh, you are feeling headache, okay, let me go and collect samples from where you are sleeping on the surface of your bed. What for? So the types of diagnosis and correct treatment will depend on the suitability of the sample you are collecting. So you have to narrow it down. That will the organism you are looking for will tell you. For example, if you are looking for fish now, you cannot say you are looking for fish and you start going into the bush. It is not possible. So what we are saying is, so you rather go to where you can find water. That's where you can find the fish. And different types of water also have different species of fishes that you can also see. So different types of sample. We also have different types of microorganisms that you can also find in there. So clinical sample like blood, urine, sputum, stool, wound, and so on. Then environmental sample, just like I said, soil, water, hair, surface swab from various locations and this. Then food sample, I've also mentioned it, from food production or consumption. Then if you are looking for animal samples or plant samples, so collected from animals or plants suspected of harboring microbes that may affect health or the environment. Okay, so how will you collect the sample? Under normal circumstance, you make sure that collection of your sample is going to be done on with sterile container you are so sure of that is not going to contain any microorganism. So sample must be collected using sterile equipment as well. It's not that you have a sterile container but you don't have a sterile equipment to collect them. So you should not introduce external microorganism. That's why you have to make sure that all your containers, including your equipment, they are not source of contamination once again. Then proper labeling. You must label your sample in terms of the time collection, location where you are collecting it, and the patient ID if you are dealing with human sample, and the type of sample. Are you talking about are you collecting urine? Indicate it there. Stool? Indicate it there. So and so on. Then the next thing that you need to know is the appropriate handling of your samples. Some samples like those of pathogen need to be transported under specific condition. There are some microorganisms that they will not survive outside their natural environment. They will not survive anything outside their environment. So you need to create an environment for them to make sure that they also survive in that environment until you get to your laboratory or the laboratory whereby you are going to start working with them. So to preserve the viability of microorganisms, I've said that. So what are the ethical consideration when dealing with clinical samples? For example, if you want to collect sample from any human, the first thing you have to seek the person's consent. The person, you must inform the person, oh, where I want to see, I want to know whether you have malaria or you do not have malaria. And in trying to do that, we are going to collect 2 to 3 ml of your blood sample, and the blood sample is going to be used for this purpose, and for this purpose, and for this purpose. So please, this is informed consent. Do you understand, or is there any question that you needed me to respond to? So before the person will now consent, the person must consent, written consent that okay, she is the one that has authorized you to collect his blood sample. It's not that you just get to a community or you just batching into somebody's somebody community and start telling them that okay, I want to collect your blood, okay, whether you like it or not. No. The person must give you an informed consent. The person must consent before you collect that sample. For environmental or food sample, ethical standard may relate to handling and disposal of samples to make sure that you do not harm others and you do not harm the ecosystem. Do not trample upon or violate the composition of the environment, that is the sanctity of that environment. Must not introduce pollution in trying to collect sample, and you are also introducing another problem. No, you must make sure that you follow all these ethical standard. Okay, so when you now get to the laboratory, that is what is going to be. So the first thing is a collection of sample followed by laboratory work which has to do with characterization. Characterization include culture and isolation. That's when you now use different types of media to start feeding. For example, now you want to rear a lion. You are taking lion from the jungle. You have to make sure that while you are transporting the lion along the line, it does not sustain injury, the lion still survive. So and when you now get what are those foods that you'll be giving to lion? For example, if you are rearing dog, its own food is going to be different from if you are rearing cats and if you are rearing lion. So you cannot just say that because cat and lion they are of the same family and that's why you also feed them the same food. No. So that is the type of carnivorous animal will depend on the media or the food that you are going to give them. So the same thing with media selection. The collected samples are cultured will be cultured on appropriate media that is going to support the growth of microorganism. So for example now there are some media that are selective. They will only allow the growth of other certain microorganism. For example now maconky will only allow the growth of gram-negative bacteria. So any bacteria or any organism that grow on maconky, you will know that it must be a bacteria number one and it must be a gram-negative bacteria. That is selective. Another one is a differential. So differential from the word differentiating. So it will help you to be able to differentiate certain microorganisms based on their metabolic activity. For example, we have some microorganisms that they metabolize hemoglobin uh, by eating or feeding on the blood, the hemoglobin in the blood because of their hemolytic activity. So differential is going to differentiate them from there. Maconky is can also be regarded as a differential because the pattern of growth of lactose fermenters and non-lactose fermenters on maconky will differentiate them the same way they grow. Hemolytic activity of certain microorganisms will differentiate them and single them out. Another word for isolation is single out. You want to single them out. You want to make sure that they are alone before you be able to study them. So you are not interested in studying the whole population. If you are interested in studying the whole population, a different approach is going to be taken. Okay, so the next one is a colony morphology. After you want, so when you now introduce them into okay, another type of media is enrichment. Why I'm not dwelling too much on that is that you will be used to that like when you start your practicals. Okay, they will tell you more about media, selective media, differential media, enrichment media. But one thing I want to tell you about enriched or enrichment media is like somebody who is taking garri. You want to take garri, so you can enrich garri is carbohydrate, but you can enrich your carbohydrate with other classes of food like garri when you combine it with groundnut, when you combine it with milk, meat and so on. So you are enriching it. That is what is called enrichment agent or enrichment media. Okay, so then incubation. Samples are incubated under optimal temperature, pH, oxygen. So not all bacteria can grow at all conditions. No, no. There are bacteria that when they want to grow, they will require that the pH should be acidic. Whereas some will require that the pH should be basic. So anything short of that, they won't even grow. They won't survive. So that is about incubation that you also needed to know. Another one is a colony morphology. Morphology is that the way you now incubate them for like overnight. If you are dealing with bacteria, if in your sample you have bacteria, when you come back the following day you will see growth. If bacteria is not there, if it is fungi, you have to wait a bit longer like between 3 and 5 days before you see them coming up. Okay, and there are some certain bacteria like Mycobacterium tuberculosis, that is the causative agent of tuberculosis. So they require certain special media for them to be able to grow. So even if you incubate them, although they are bacteria, but they are slow growing bacteria, they are slow growers because of the presence of certain mycolic acid in their cell wall. They are slow that even sometimes you might not even see growth until maybe after 8 to 10 months. I mean 10 weeks before they will now start coming up. So that's why I said depending on your organism of interest. So depending on your organism of interest, so you have to take note. So we're going to stop in the next few minutes. That is the next 5 minutes. We are going to stop, and wherever I am able to get into today, we will continue from there in the next lesson. Don't forget what we are trying to do. What we have been saying this morning is that we are talking about characterization, isolation, and classification for you to be able to treat microorganism correctly. I said, I told you about different types of terms and terminologies that you will come across whenever you are studying or whenever you are studying discord, which is microbial evolution and ecology. I told you something about horizontal gene transfer, that is exchange of genetic material between uh, microorganism, which can either be the same species or different species as well. Then another one is vertical. Vertical, I said you are drawing from up to down, that is from parent to offspring. So those are the things that will actually help you to be able to understand and differentiate between horizontal colleagues. For example, now if you exchange money, maybe you are hungry and your friend decides to just give you money, that is horizontal money transfer. But if the money is coming from your parents to you, that is vertical. That is vertical. So the same thing with microorganism, the same thing with bacteria in particular. So if the exchange of genetic material is coming from the parent to the offspring, that is a vertical gene transfer, VGT. But if it is a horizontal, that is their colleagues, they are at the same level. That is from friends to friend, colleagues of different, I mean of the same, it could be of the same age group or might not even be of the same age group. That's why we said uh, horizontal gene transfer is the transfer or the process by which genetic materials are transferred from uh, from one microorganism, which could be of the same species or of different species. So that is a horizontal gene transfer. Another terms that we have also mentioned today is a transformation. Transformation, when you have an exchange of genetic material from the environment, the DNA is free, and the recipient DNA, the recipient bacterium has decided to just do what? Take it up. That is a what? That is transformation. Free, dead. When microorganism dies, Yeah, when bacteria dies, is going to die, but the genetic material, the DNA, is still going to be in the environment. So the taking up of another person, that is inheritance. You are inheriting another person. For example, now we also have a transformation. We can also explain it as in some culture, Nigerian culture. Just see that if the owner of any wife dies, it is the senior brother, I mean the next person that they will will it to. So that is like transformation. Then another one is I also mentioned conjugation. Conjugation from conjugal bliss, sexual reproduction in bacteria. So is as a result of conjugation. Okay, and I told you that bacteria can also be differentiated into male and female. Okay, so when a recipient cell come in contact with a donor cell, they will now exchange their material, genetic material, in form of a conjugation. And I also made mention of transduction. Transduction, anytime you come across the word transduce, that is from phage, from bacteriophage, from a virus to the bacteria. That is when there is transfer of genetic material from the virus to bacteria, and the genetic material of the virus will now become incorporated or integrated into the recipient's DNA, and it will now continue to replicate along with it. That is transduction. And another thing that I've also mentioned in this lecture is I mentioned something about mutation. I said mutation is a sudden change, sudden alteration, sudden manipulation, sudden transformation in the DNA sequence or in the genetic material. So when I told you that evolution cannot occur if there is no mutation, so mutation can be spontaneous or it can be induced. This is not the lecture we are going uh, to talk more about mutation. We are going to discuss more on that. Then I also made mention of a genetic recombination. Recombination, that is when the process by which genetic material is shuffled between different organisms, So that increases what is called the diversity, genetic diversity, so which is very very powerful for evolution to also take place. I also made mention of antibiotic resistance. I said antibiotic resistance is as a result of that there are some bacteria or there are some microorganisms that, apart from their DNA, apart from their main DNA, they also have another thing that is called plasmid, which is extra chromosomal. And wherever you see plasmid, you should know that what are they using plasmid to do? They are using it for drug resistance. They are using it for antibiotic resistance. So we will know more about all these as we proceed in this course. Don't forget that I am still treating you as a newcomer, as very very novice in microorganism, and that's why I mean sorry in microbiology, and that's why I'm trying to take my time to explain all these words for you, so that you'll be able to know them one after the other. And apart from that, I also talk about collection of samples. How can you collect samples? That the type of organism you are looking for will determine the sample you are going to collect. So if you are looking for, I cited an analogy. We cannot be looking for fish and decide to head to the bush or head to forest. No, you have to go to where there's going to be water. So the same thing with sample collection. The organism you are looking for will determine the type of sample you are going to collect. If you are looking for microorganisms that are found in urine, and you are collecting blood, you know that it is inappropriate. So samples could be clinical, it could be environmental, it could be food samples, it could be animal, or it could be also what? Plants. So then collection of samples, collection method, you should make sure that everything, both your equipment, um including your containers, everything you are going to use in collecting your samples must be sterile, must not introduce contaminants. So you need to know that as well. And apart from that, I also told you that one of the characterization of your microorganism will now depend. So the organism you are looking for will give you an informed decision of the type of media, the type of food you are going to prepare. The type of visitors you are going to have will depend, um you are going to have will now direct you that what kind of food should I prepare for this person. So if you want your visitor to have a very good time with you, that means you must prepare the favorite food of that person. So the same thing, and I cited the example of media. I said we have selective media, we have differential, we have enrichment. I told you selective. Uh, McConkey agar is selective agar that will only allow gram-negative bacteria to grow, no other, only gram-negative. So, and I also said, made mention of a differential enrichment as well. And I also talked about incubation, that is the type of growth, the type of microorganism you are looking for, or you are dealing with, or you are handling will depend on the incubation period. If you are dealing with bacteria, that means between 18 and 24 hours, you should be able to have a an appreciable growth. And if you are dealing with fungi, I said that between 3 and 5 days, that's when you'll be expecting them. And I cited another example, clinical example of Mycobacterium tuberculosis, whereby for you'll be expecting the growth to be to start from 8 to 10 weeks. So that's why I said the type of microorganism would depend on the incubation period. There are some bacteria, or there are some microorganisms that they need high pH for them to be able to grow well. There are some that are aerobic, there are some that are anaerobic. There are some that oxygen, the smallest quantity of oxygen will be poisonous to them. So that's why you need to also know the incubation period of the organism you are looking for. um Incubation period. Look at when they will perform optimal. So just like I said, I'm going to leave a link later on the day. I'm going to leave a link in the comment section so that everybody is going to have an opportunity of participating in the tests, in the assessment we are going to do. So once again, I want to really thank you for your attention this morning, and our lecture is going to also continue as we plan it. Don't forget that this is the voice of Ms. Adeoti Olatunde Michael. I'm a lecturer of microbiology, and this course is microbial evolution and ecology. So, once again, I want to thank you. I want to see you. Don't forget to comment, like, and also make sure you also share the link to your colleagues who are not even here, and also the link to help us